Hi guys, this is Mrs. Allgood again. I'm here to teach you about plant cells and tissues today. So I figured we would start off with a little bit of an overview. If you remember your levels of structural organization, you'll remember that, atoms make up molecules, molecules make up cells, cells make up tissues, tissues make up organs, and organs make up organ systems. So what I'm hoping is that over the next few days, you'll be able to relate those terms to plants, and you'll understand that plants have specific types of cells, and they have specific types of tissues, they have organs, and they have organ systems. So I'm just here on this. This is a sheet that was a single sheet that you got when you picked up all your papers. Okay, so first, these are some of the different cells that we're going to learn about today. Okay, so we're going to learn about parenchyma, collenchyma, sclerenchyma, tracheids, vessel elements, and sieve tube members. So take a second to get those written down. And again, whenever you need to pause the video, just feel free to. I'm going to keep moving. The three types of tissues that we're really going to focus on today are going to be dermal tissue, ground tissue, and vascular tissue. So dermal tissue, you can imagine, has to do with the plant coverings, like an epidermis, okay? Vascular tissue, you already heard about that. That's going to be things like your xylem and your phloem. Ground tissue, for what we're going to talk about today, is kind of like a filler, but we'll talk more about that tomorrow. Okay, your three major plant organs are roots, stems, and leaves. So tomorrow, I believe we talk about some modified roots. This will be Thursday's lecture, so I guess early next week, we'll talk about modified stems and leaves. All right, and then organ systems. Okay, a root system and a shoot system. So that'll be a couple days away as well. But this is just to kind of put things in perspective here. So today, our focus is on the left-hand side. We are focusing on plant cells and tissues. So cells. What we're going to be filling in is your long sheet. You should have an 11 by 17 sheet. I believe cells are all on one side, and then tissues are on the other. And then on Friday and maybe Monday, Tuesday, we'll be talking more about roots, stems, and leaves. Okay. So in order to fill this in, you've got to find your long sheet, your 11 by 17 sheet that has plant cells on one side and plant tissues on the other. So find box number 1. Let me find my long sheet as you're finding your long sheet. Okay, find box number 1. That's going to be in the upper left. That's your parenchyma cells. Okay, this is the most abundant type of cell in most plants. I'll tell you some different places where it'll be located. One thing you're going to hear a lot about today in notes is whether these cells are alive at functional maturity. So these particular ones are alive, but some of the other ones will be dead at maturity. Those are usually found in your woody stems and tissues. Parenchyma cells are quite flexible, so they only have primary cell walls. Notice here you do see it kind of looks like an empty looking cell, and it's not empty at all. It just has a very large vacuole for storage. So some of the jobs of parenchyma cells, this is going to be where photosynthesis occurs in the leaf. So if you think back to when we learned about the light reactions and we were learning about where in the plant photosynthesis occurs, you did see a picture that was kind of like this. You at least knew that you have palisade. Now, we didn't call it palisade parenchyma. We called it palisade mesophyll cells. If you remember that term mesophyll is where photosynthesis, those are the kind of cells that photosynthesis occurs in. Well, mesophyll cells are actually a type of parenchyma cells. So if we didn't want to call it palisade mesophyll, we could call it palisade parenchyma. And then the cells that are a little bit more spread out on the bottom, those would be spongy parenchyma, or like you learned it before, spongy mesophyll. Okay, parenchyma cells, like we said, they have these large vacuoles for storage. And so this is going to be when plants store away excess sugars as starches in their roots. This is going to be where plants store away those excess sugars. Parenchyma cells are also what makes up the fleshy tissue of most fruit. So if you like eating peaches or apples or pears, when you bite into that fruit, the fleshy part that you're eating is most likely parenchyma cells. Okay, so we're going to go to the box right below that, box number 2. This is your second type of cell. This is collenchyma. All right, these also lack secondary cell walls. They have primary cell walls, but they are unevenly thickened, which means some parts of their cell wall will be thicker than others, and that seems to be very characteristic of collenchyma cells. They tend to occur in strands or strings. Okay, so when you think of like a celery string, like when you eat celery, those are collenchyma cells all linked together. So their job, collenchyma cells are really important at providing support in the parts of the plants that are still growing. So if we take off whatever I've drawn in there, okay, your collenchyma cells are going to be right about here. These little circles that I had drawn over, okay, these little circles here, those are your vascular bundles. So if you remember, if you were in 3rd or 4th block and you did that monocot-dicot coloring sheet, you might remember seeing some of these little vascular bundles that have xylem and phloem. That's not what we're talking about with collenchyma cells. Collenchyma cells are going to be more external, and they're going to create those strings that are very characteristic of celery. Okay, next let's look at sclerenchyma cells. Okay, so that prefix sclere means hard. And so these cells are going to be much more rigid than your parenchyma or collenchyma cells. And one of the things that makes them so rigid is that they have secondary cell walls. Remember, all plant cells have at least a primary cell wall, but then some plant cells will also have a secondary cell wall. And this is one of those cell types. These cells are hardened with a substance called lignin, which is one of the chemical components of wood, and that's going to be very important for reinforcement. That's also going to contribute to their more rigid structure. So sclerenchyma cells only occur in regions of the plant that have stopped growing in length. Again, with these cells being more rigid, it wouldn't make sense for them to be in the parts of the plant that are still growing. And when mature, most of these are dead. Okay, and again, that's because of that lignin, that's because of that secondary cell wall. So there are 2 types here. So we're going to go into the 2 types of sclerenchyma cells. Okay, first up is this one. These are fibers. So these are long and slender. They're tapered at the ends. And if you ever hear that term tapered, it means they get skinnier at the end. So if I was going to draw them, they look kind of like this, okay, where they would get skinnier at both ends. So needle-shaped pointed tips. These tend to occur in bundles or strands. So if you think about when you hear the word fiber, what you tend to visualize, that's what these are. And the example would be something like hemp fibers that are used to make rope. Okay, let's look at the other type of sclerenchyma cell. Okay, these are sclerids, or they're also known as stone cells. So they have very hard cell walls, and we find them mainly in two places. They make up the gritty texture of some fruit, like a pear, how it has more of a gritty texture when you bite into it. And then they also make up nut shells and seed coats. So that kind of goes along with that name sclera, that hardened kind of appearance. Okay, so sclerids and fibers are two subtypes of sclerenchyma cells. All right. Next, if you find box number 4, we're going to be talking about two types of water-conducting cells. So both of them have rigid secondary cell walls containing lignin, and these are going to both be dead at maturity. If it transports water, you just have to remember that it's probably going to be dead at maturity. And I know you think, well, how can something that's dead still transport water? We have a lot of things that transport water, like, that use capillary action or use other forces. It doesn't really matter that the tissue itself is dead. Water can still be transported, okay? Just going to go back a second. So if it's got lignin in it, that's a good clue in that it's probably dead at maturity. So sclerenchyma cells were dead at maturity. They had lignin. Your water-conducting cells are going to contain lignin, so they're dead at maturity. So let's look at the two types. Okay, so this one's going to be your one on the left. These are your tracheids. So the two types of water-conducting cells are going to be your tracheids and your vessel elements. So we're going to start off with tracheids. These are long cells with tapered ends, which I know is a lot like how we describe the fibers, but I'm gonna give you a way to tell these apart from fibers. Okay. These are found in the xylem of nearly all vascular plants. But this is an older type of cell, so it's going to be pretty prevalent in your ferns and your gymnosperms. Okay, notice I'm not listing mosses here. Mosses lack vascular tissue, so they are not going to have water-conducting cells or sugar-conducting cells for that matter. So you do kind of see this shape, the same sort of shape where it gets skinnier on the ends, but you might notice all these spots on there, okay? And those are not just spots, those are pores, okay? And so because these are all sort of lined up next to each other, the water can be drawn up and then can enter a different tracheid by passing through one of these pores when they're right next to each other like that. So this is the older of our two water-conducting cells. So these cells are not connected end to end. They are right next to each other, and like I said, when these little pores, these little holes line up, that's how water can get from one cell to the other. Okay, so you might have water coming up here, and then maybe it's going to pass through one of those pores and through here, and then keep going up. Okay, that's kind of how water is going to move in a tracheid. Okay, so those thinner parts of the cell wall, like I showed you here and there, where the little circles are thinner parts of the cell wall called pits, and that's where water is able to transfer from one tracheid to another, and that's on its journey typically up the stem, because we know water is going to be taken into the plant through the roots. So this picture down here is just showing you a better zoomed in picture of all those little pits. Okay, that's very characteristic of our tracheids. Okay, then we have vessel elements. Okay, now vessel elements are going to be wider and shorter, so the individual cells are very cylindrical. They really don't get very skinny on the end. And so they're wider and shorter than our tracheids. Now this is our newer water conducting cell. So whereas the tracheid is mainly found in our ferns and some gymnosperms, this is going to be found in most of your angiosperms, a few gymnosperms, and a few of your seedless vascular plants, which are going to be your ferns. Okay, these cells are connected end to end. And you can see that they've sort of got this little plate-like appearance there at the end of every cell. And these are just kind of open and empty. They allow an open pipeline for water transport. And that's just going to make transport more efficient. Okay, so keep in mind that you've got tracheids and vessel elements are your two water-conducting cells. All right, guys, we're going to move on to our fifth type of plant cell, and that is a sieve tube member. So a sieve is like a strainer, kind of like a colander that you might strain the water from when you make pasta, you know? You let the water drain out the bottom. That's what a sieve is. It's kind of like a filter or a strainer. Okay, so sieve tube members are sugar-conducting cells, and they are alive at functional maturity. So think about your two cells that conduct water, your tracheids and your vessel elements. Those are going to be dead at maturity. Sieve tube members and companion cells that go along with them, they're going to be alive at functional maturity. So these have thin primary cell walls. They're not going to have secondary. Remember, if it's more rigid in structure and it has a secondary cell wall, it probably has lignin, and if it has lignin, it probably means it's dead at maturity. So these do not have a secondary cell wall. They are not dead at maturity. They are alive. Okay? The ends of these cells, the reason they're called sieve tube members is because they have these sieve tube plates or sieve plates at the end. Okay? Through which sugars and other compounds can pass. Now, one of the things I'll show you with this is the reason that we have that is to sort of regulate the transport of sugar. So it doesn't, usually it's going to be made kind of more in the top of the plant, like by the leaves, and then it's going to travel down to the other parts of the plant and maybe to the roots. The sieve tube member, the sieve plates rather, are going to just kind of slow down that transport of sugar. It's going to regulate it. Now, on each side of the sieve tube member is a companion cell, okay? And this is kind of neat. So when you look at that sieve tube member, you don't see a nucleus, you don't see organelles. And so you might think, oh, well, they're just not showing them. No, they're not there. Because this type of cell is specialized for transport, it basically doesn't have any of the organelles that most cells do. The companion cell, however, does. I know it looks like the companion cell just has a nucleus, but it has the other organelles that you might need. And so it basically kind of does the work, does the metabolic things for the sieve tube member, and then passes that stuff on to there. But this cell is nice and open so that sugar can be easily transported through it. Okay, So let's take a look. So there are numerous plasmodesmata, remember we learned that term in the fall, that connect the companion cell to the sieve tube member. And the companion cell, even though it's not showing you the rough ER and the Golgi and the mitochondria, the companion cell makes proteins for the sieve tube member, and then those proteins can be transported into the sieve tube member through the plasmodesmata. Okay, so sidebar, sieve tube members lack a nucleus, ribosomes, vacuole, and any parts of the cytoskeleton. And that just allows more food or sugar to pass through. And so you'll find that, the AP exam the AP folks really want you to understand the relationship between structure and function. So when we have cells that are specialized for transport, like a sieve tube member, or like your red blood cells lose their nucleus at maturity, and the reason that they do is so that they have more room to bind up oxygen and transport oxygen throughout your body. Okay, same thing with the sieve tube member. The reason that the sieve tube member looks so empty is so it has more room to transport sugars. Okay, so those are your 5 main types of plant cells. And then we had some subtypes, but we had parenchyma, collenchyma, sclerenchyma, and then underneath sclerenchyma, you had fibers, and then sclerids, or stone cells, which is what made up the gritty texture in your pears. And then we looked at our water-conducting cells. Remember that those are your tracheids and your vessel elements. And then we looked at sugar-conducting cells, which are mainly the sieve tube members, but you need to know that the companion cells help them out with a lot of their metabolic processes. Okay, so you can flip your sheet over. We're going to start over on the left. Okay, these are 3 main types of plant tissues. So the front side was all about main types of plant cells. This is all about main types of plant tissue. So first up is dermal tissue. So when you see that root derm, you kind of think skin, you kind of think a covering tissue. So this is the plant's outer protective covering. And you want to think of it as being analogous to our skin. So it's there as defense against pathogens and other things that can cause damage. And so just like our skin has an epidermis, so if you've taken anatomy, you learned all about the epidermis and the dermis. Your dermal tissue, your plants do have an epidermis as well. And cells of the epidermis are what actually secrete the waxy substance for the cuticle. And remember that that cuticle is going to help prevent against water loss. So when you look at this, here's your upper epidermis. Let's say if this is a leaf. This would be your lower epidermis. This is your palisade parenchyma. This is your spongy parenchyma, or you can call it spongy mesophyll. And then the cuticle is going to be outside of that epidermis, okay, because it's these epidermal cells that are secreting that waxy substance for the cuticle. And so there's your cuticle down here as well. All right, 3 types of specialized epidermal cells. First are root hairs. Okay, root hairs increase the surface area for absorption. They are not lateral roots. Okay, the only time you're really going to see root hairs is if you were to germinate a seed yourself, like soak it in water overnight, wrap it in a paper towel that's damp. If you were to germinate a seed yourself and you watch it come out of the seed coat, you might be able to see a root hair. But if you just go find a plant outside, chances are it's not going to have root hairs. It's going to have lateral roots that are going off to the side. So root hairs are something that increase the surface area for absorption, and they are very evident in like an embryonic root or in a germinating seed. Trichomes, this is what we counted in our plants in lab. These are hair-like outgrowths from the epidermis. There are many different trichomes in different plants. They don't all look the way that you saw them in lab. Some can secrete sticky fluids or toxins to defend the plant against insects or herbivores. Okay, so that's the purpose of having a trichome. It is a defensive structure. The third type of modified epidermal cell is a guard cell. Okay, and the guard cells surround the stomata. So the stoma, that is one hole, one opening. Stomata is plural. Unless I got that backwards, but I don't think I did. The guard cells guard that opening. So this is a guard cell right here, and this is a guard cell right here. So when we talk about transpiration, probably next week, we'll look into that a little bit more. But guard cells surround the stomata, and they are modified epidermal cells. So all those three cells that we just talked about, in addition to the cuticle, those are all modified from the epidermis. All right, ground tissue. So first, let me have you fill in your blanks there. This is something that we'll look at a little bit more closely tomorrow and the next day, or tomorrow and next week. Because I think tomorrow we talk about the pathway that water takes through the root. So what you are looking at is a cross section through the root. And these are some of the key structures that you might see. So the center of that, where you're just looking mainly at parenchyma cells, that's called the pith, okay? Epidermis is the layer of cells on the outside. The cortex is in between the epidermis and the endodermis. And then these little circles, you have big circles and little circles, that's your xylem and phloem. So you're going to get better exposed to that a little bit tomorrow. Okay, so your cortex is basically the filler ground tissue between your epidermal tissue and the vascular tissue, which is your xylem and phloem. And then your pith is what's internal to your vascular tissue. You can kind of think of it like the core of your root. Okay, the pith is what's at the center. So we had epidermal tissue, which was a covering tissue. We have ground tissue, which is basically kind of like a filler. And then we have vascular tissue. Now, I know you've heard these terms before. You're going to hear about them here and there throughout this unit, just to get you more familiar with them. So this is vascular tissue. You've got your xylem. Xylem conducts water typically from the roots to the shoots. So from the bottom of the plant upward, typically. And the plant cells that make up your xylem are going to be your tracheids and your vessel elements. So remember, it kind of depends on what kind of plant we're talking about as far as which one it has, because one is older than the other. If you go back to look at your notes from the front side. Phloem, okay, phloem, I think that PH sound is kind of like an F sound, so phloem is for food. Phloem transports sugars typically in the opposite direction. Those sugars are being made in the leaves and it's being transported downward, maybe to be stored away in the roots or maybe to some other parts of the plant. It might be going out to be deposited where fruit is forming, because we know fruit is going to have sugar in it. So it may not be going down to the roots. It may be going to the fruit as well. And so sieve tube members are going to be your main cells that make up phloem. But remember, they can't do their job without companion cells there, because companion cells are making their proteins and carrying out all the metabolic reactions that those sieve tube members need. Okay? So it looks like that's it for notes for today. I'm trying to do this a night early just to get a little bit ahead, so I will go and post it, but these really are your notes for Thursday. If you are seeing this in the morning, please make sure you go ahead and do that quiz on Google Classroom just to get that out of the way. I'll be making another one and posting another quiz shortly, so you want to keep up with those. Thanks so much. I miss you guys. Hope to see you soon.